All right, everybody, let's get started. I'm not going to do any lengthy introduction, except to say that Yanya is in Dudley Laming's lab and is working on this really cool stuff about exactly how diet interacts with outcomes and a little bit with TOR, which is my favorite. So take it away.
All right. Hi, everybody. My name's Young, and thank you for the introduction. And I've been a postdoc with Dudley in the Laming lab for about 5 years. And today I'm going to show you guys what I've been working on for the past probably 4 years or so. And this is a story about a diet-drug interaction between a dietary intervention that we've been working on and also a drug that we sort of been focused on. But first, I sort of want to introduce this idea of diet-drug interaction by introducing an example of it that is a little bit more familiar to people in here, which I assume everyone here lives in Wisconsin. So if I could advance the slide. So this example has to do with cheese and depression. So I came across this pretty dramatic statement in this paper, which says that no deaths has ever been documented with foods other than cheese. And being from Wisconsin, I thought this was pretty fitting for this talk. So we're just not talking about any kind of cheese. Specifically, this is the good stuff. I don't know if anyone here had it because it's quite expensive, but you can get these 20-year-old cheese. They're old enough to vote. But if you look pretty closely, there are these gritty textures on it, and they're a little crunchy when you bite into it, and they're just delicious. And these things are actually tyrosine crystals. And as the cheese gets older, they become tyramine crystals. So just keep this in mind. So the drug part of this story comes to antidepressants. So in the 60s, we developed the first line antidepressants, which are the monoamine oxidase inhibitors. So monoamine oxidase is an enzyme that degrades molecules such as dopamine, so that if you have a synapse that has too much dopamine in it, the oxidase is going to degrade them so that they don't have extrasynaptic activity. That's all good and all, but if you have low dopamine signaling, as you here, the monoamine oxidase is not really helping, which is why we develop these inhibitors that block the activity of the monoamine oxidase, which increases dopamine activity, and you'll feel a little bit lively again. So it turns out tyramine, that cheese molecule that I mentioned a little earlier, it's also degraded by monoamine oxidase. So this is called the cheese effect or the cheese reaction. If you were on these antidepressants and you were eating a lot of these aged cheese, you're going to have a buildup of tyramine. And what that does is that it displaces all the noradrenaline in terminals, and it causes an increase in blood pressure. And that's going to lead to a hypertensive crisis. So over 500 cases of this has been reported, and more than 20 people have died. But this is from the 60s, so a long time has passed, and we don't use this kind of antidepressants anymore as much. But I think this is a very good case of illustration of why diet-drug interaction is important, if not to understand the underlying mechanisms of either the diet or the drug, and also just for safety reasons. So we're going to take a little bit of a step back and try to see that in all foods, there are other foods besides cheese. Most foods, or all foods, are containing micro and macronutrients. And today we're going to focus on dietary protein, which is a class of macronutrients. I'm sure everybody here knows that proteins are multimeric compounds that work as the workhorse of a cell, and they're made of these subunits called amino acids. So this is going to be important later on. There are about 20 most common amino acids, and that includes tyrosine, which was the cheese component I mentioned a little earlier. And if you were to ask popular media, let me move some of the Zoom things. if you were to ask popular media how much protein you'll be consumed, you'll only really get one answer, and that's to eat more. So this is sort of, it's much easier to sell more stuff than to sell less stuff, or you can make money that way. So everything you see online is going to tell you to eat more protein when you're a kid, when you eat protein when you're middle age, and eat more protein when older, just more. And I thought some of this got pretty ridiculous because eggs were pretty expensive. Like, what protein should we get to replace them? And then eventually it was just, how do you put protein in the food that has no protein in it? So, but what does the actual research say? So we do have a lot of retrospective epidemiology data. Also, work done by Morgan Levine when she was a postdoc has shown that in these graphs, which are time to mortality graphs, you can see that in the middle age, about 50 years of age, it appears that you get a benefit if you consume less protein. But you can see these lines cross over, which means that in the later age stages, in 70 to 80 years of age, it seems that more protein is beneficial. So in these graphs, it's sort of kind of hard to tease out because the effect size is very small. But what was not small effect size in the same study was a correlation between dietary protein and diabetes. So the top line on the left, you can see that clearly people that are eating a low protein diet receive a benefit from protection from diabetes and also from diabetes-induced mortality. From the statistics, we can see that people who are eating a higher protein diet have 4 times more risk of developing diabetes. And this is true for all age stages. As you can see, the lines really don't cross, which means that low protein diet across all stages have some kind of benefit from diabetes protection. So the next question we want to ask is, how does this contribute to lifespan and also health span in the long term, which means that we turn to mice because they only live about 3 to 4 years. And here, work done by Crystal Hill has shown that if you feed mice a low protein diet for a lifetime, you're able to extend their lifespan. So you get a little bit of lifespan extension effect in these animals. So for the past 10 years or so, the Laming Lab has been trying to figure out how does the low protein diet elicit its benefits. And remember, I showed you there are about 20 common amino acids. And of these 20, 9 are essential, which means that our body cannot make it. You have to consume it. And of the 9, 3 of them are sort of a special interest for our group, and they are called the branched-chain amino acids. So these are leucine, isoleucine, and valine, so named after their similar structures. So BCAAs are very interesting to us because many groups, including Christopher Lynch, Sean Adams, and their colleagues have shown that they're associated with many diseases. In case here, the circulating level of branched-chain amino acids are highly elevated in people that are insulin resistant and also in rat and mouse models as well. So we want to ask the question if we can improve the symptoms of these people, of these animal models, if we're able to take the branched amino acids out of the diet. So how do we study this? The way we approach this is through something we call the amino acid defined diet. So basically there are these, all the food has a protein profile of the amino acid breakdowns. And we simulate that by making a food pellet made completely full of amino acids instead of proteins, but making it so that it has the same profile as the complete food. So we call this the amino acid defined diet. And the reason we do this is so that we're able to access and manipulate individual amino acids. So the goal here is to take the branched amino acids, decrease everything about 66%, and then we increase the non-essential amino acids a little bit, just so that the macronutrient ratio between protein, carbohydrate, and fat remains consistent. Okay, so we have this low branched amino acid diets. And one of the experiments that we did, Nicole did when she was a graduate student in the lab, was that she showed this low BCAA diet is able to extend lifespan as well as a low protein diet. So the next question you would ask to get closer to the mechanism is which of the three branched amino acids, leucine, valine, or isoleucine, are mediating these effects. So we went on to make a diet series that are low in one specific branched amino acids. So you can see here, we have low leucine, low isoleucine, and also low valine. And we started to phenotype the animals. So this is the glucose tolerance test. So we performed this test as part of evaluation for metabolic health. So I know a lot of you are not that into metabolism, but in this test, we inject the animal with glucose and we see how well they're able to maintain their blood glucose levels and how well they're able to bring it back down to zero, to their baseline, which the time graph you can see on the left is when the beginning of the injection. So you can see that if we calculate the area under the curve of these animals, the lower would indicate that I had better glycemic control. So what's very interesting is that the animals that are fed the low leucine diet do not have any benefits from their dietary intervention. So they have the same area under the curve as the control, while the animals that are fed the low valine diet, and especially the mice that are fed the low isoleucine diet, have a dramatic improvement in their ability to maintain a low blood glucose level. Okay, so we sort of focused on the isoleucine for this purpose of this talk, and work done by Cara, who was a previous postdoc in the lab, and she's opening up her own lab in the University of Bath. I heard it was pronounced Bath if you're posh. But she showed that if you fed animals a low isoleucine diet for a lifetime, they have a lifespan extension. And we've always known that if you have caloric restriction as part of a dietary regimen, you can have extended lifespan. So here she's showing that this is not the case with our dietary interventions. You can see the animals are eating at least as much as the control group, and in many cases, they're actually eating more. So what I've shown you so far is that we're getting more and more specific in our dietary interventions in our attempt to try to understand how these diets are eliciting their benefits. So I didn't show you all the phenotyping data. There are some very consistent phenotypes I want to point out. Most of it relates to an improvement in metabolism. So you can see that animals usually have weight loss and they're more lean, so much less fat mass. They have improved glucose tolerance that I've shown you. And they also have increased energy expenditure. Okay, so I showed you earlier that there might be this crossover effect where it's the optimal dietary protein intake is age dependent. So a reasonable question to ask is whether or not these diets could benefit mice in old age. So this is one of the questions that I pursued as a postdoc. And the way we answer this, we take animals that are raised on child diet at NIH, and we get them at about 20 months of age, and they will be roughly equivalent of 60-year-old in humans. And we put them on these dietary interventions, and we see how it affects their physiology. So first, I just want to start by saying that they also eat more food, equal or more food than the control group. So it's very important for the elderly that they don't lose their appetite, because that is very detrimental for health when you're at a stage that you can't really build muscle. So despite eating more, these animals have decreased body weight. The blue line is the body weight for the animals that are fed the low amino acid diet, which is the defined version of the low protein diet. And the orange, it's the animals that are fed the low isoleucine diet, which means that they lose even more weight than animals on the low protein diet. This weight loss consists of both fat and lean mass in the animals that are fed the low isoleucine diet. But the low protein fed mice only have lean weight loss. if you do the glucose tolerance test again on these animals, you can see that both of these dietary interventions are able to improve their glycemic control. And this is the energy expenditure data. On the left, you have energy expenditure over time. You can see both groups of animals that are on the diet have an energy expenditure that's above the control group, especially in the dark phase. On the right, it's usually accompanied by the spin data, which means that we average all the data between the light and the dark cycle, light or the dark cycle together to get an average for statistical purposes. So overall, I've shown you that these diets enhance metabolism in H mice. And we also do a lot more specific profiling in different organs. And for this talk, I'm going to show you a little bit of protein and gene expression data in the liver. So here we did a transcriptomic analysis in the liver. So initially, we asked the question of how aging affect the gene expression profile in the liver. So we're able to identify about 600 genes in the males and about a lot more, about twice more than that in the females. And these genes are both up and down regulated, causing by aging. And if you take these genes specifically and you ask the question of how the diets affect these genes, you're able to see that most of these genes are sensitive to the diet. So it's not coming out very well on the projector, but the outer rings are the genes that on the inside, except that they're altered by the diets. And a very small proportion of these genes are not sensitive to the diets, and they remain in the same direction as they were driven by aging. So if you take these genes that are sensitive to diets, then we do an enrichment analysis, you're able to see that many of them are associated with metabolism, which including terms such as thermogenesis, cellular respiration, and also mTOR signaling, which I'll get into a little bit more later on. And they also correspond to very specific promoters, which we're now trying to target as sort of a therapeutic approach and also as means to understand the mechanism of the diets. So these are gene expression data. We're trying to understand things at a protein level as well. Rich Miller from the University of Michigan have been trying to understand aging from a different perspective. And you often hear a lot about biomarkers of aging. And from the way he explained it, was like biomarkers shows how aged you are on the molecular level. But he's come up with this concept of the aging rate indicators. So he's able to compare all the dietary interventions also and drugs that extends lifespan. And he saw that they all have a shared phenotype or molecular phenotype. And he believed that these are indicative of the speed of aging rather than how far you've aged. So this is a panel of a lot, like 30 or 40 proteins. I only want to show you a couple of them. So I mentioned a little bit earlier, mTOR signaling is altered. Here you can see in the aged animal, mTOR expression or activity is highly elevated, and this is decreased by both our dietary interventions to control levels. And this is also true in MAP-ERK signaling as well as lipogenesis genes or lipogenesis genes. And this is especially suppressed by the isoleucine diet, low isoleucine diet. So what I've shown you so far in sort of this section was that you have these sort of phenotypes that are associated with a beneficial dietary intervention, that you also see them in the aged animals. And you also see a youthful molecular signature. But of course, there are also downsides with these diets as well, and that include the loss of lean mass. And some data that I haven't shown you that's in the publication is that there are some changes that were significant as far as muscle function and also heart function, but we're not sure if they're good or bad because they do not correlate with either the young animal or the old animal. So it's sort of something that we're still working on understanding. So I sort of talked about diets for like 15 minutes. So you're probably wondering where is the drug of this diet-drug interaction? So don't worry. The NIH has been working on drugs for a really long time, especially the National Institute of Health or National Institute on Aging. And they have this interventional testing program where they put small molecules into or in compounds into food for mice. And they do a very standardized protocol to test lifespan in mice. So these are the initial list that they've come up with that they've tested. And it's been like 20 years, so they've come up with a lot more molecules. And of this cohort of molecules that are tested in ITP, one of them is rapamycin. And this is sort of our favorite drug. And rapamycin was discovered initially in bacteria samples, just literally in the dirt of Easter Islands. And that's sort of how it was named, where it was named after. And in these lifespan extension studies, rapamycin found to extend lifespan in males and females. And if you start the animals, whether in early life, midlife, or late life, and it also extends lifespan if you transiently give it to them for about 3 months, and the drug wash out about 2 weeks, but they still get a lifespan extension effect. And there are also ongoing Alzheimer's disease trials looking at the effects of rapamycin. So this is one of the most promising drugs that we've been studying. So because there has been so much spotlight on rapamycin, we do understand how rapamycin works. So it inhibits mTOR complex 1 and also complex 2. So these two complexes have the same core component, that is mTOR. But complex 1 is defined by its Raptor binding partner, while Richter is the defining binding partner for complex 2. So what's very interesting is that acute treatment of rapamycin would inhibit complex 1, while complex 2, you need to give it the drug chronically before complex 2 is inhibited. So how it works from a pharmacokinetics perspective, I thought was pretty interesting is that at least complex 1 is pretty simple, where rapamycin is able to bind mTOR and displace Raptor. But in the case of complex 2, Richter actually covers up the binding site for rapamycin, which means that it can't be inhibited. But as the protein turnover and you give the drug chronically, it's able to access newly expressed, newly translated mTOR, meaning the drug getting there before Richter can bind, causing the complex to not be able to form properly. And that leads to the acute and chronic discrepancy between complex 1 and 2 inhibition. So why does this matter? What's interesting is that the inhibition of chronic inhibition of complex 2 leads to glycemic dysregulation. And this is mostly associated with a disruption of insulin signaling. So if you do a glucose tolerance in these animals, you have glucose intolerance, so that the glucose level of these animals shoot up and they're not able to bring it back down to baseline easily. So remember, I just showed you that animals that are on the isoleucine restricted diet, it's able to improve their glucose tolerance. So we sort of had this, I guess, brilliant idea at the time, but you'll see why it's sort of surprised us. Is that we can put both of this drug together, or both of these treatments together. So you have a mouse that's on a diet that can extend lifespan. And we also treat that mouse with a drug that extends lifespan. And hopefully that you get a super healthy mouse, because the diet will improve the metabolic health of these animals and cover up the side effects. So we have a combination treatment. So this is the experiment, and the blue line would always be the mouse that is on the combination compared to the orange, which is the diet by itself. Mouse loses weight as you've seen before, but what's really interesting, and you can't really see that well on, on this projector, is that when you give these isoleucine restricted mice rapamycin, it completely blocks the weight loss effect of the diet. So the drug wins out on all fronts as far as body weight goes. If you look at the glucose tolerance of these animals, they still have glucose intolerance. So that it's almost if they're eating the controlled diet. If you look at the energy expenditure, this is also true. So rapamycin completely blocks the energy expenditure increase induced by the diet. Okay, so we sort of want to first look at this diet-drug interaction in a more realistic setting. So in terms of mouse physiologists that ask that question, it just means like what happens with the mouse's fat. So we feed the mouse a Western diet. Western diet is high fat, high sucrose, also contains a lot of cholesterol. So we precondition them for 3 months. You can see that mouse steadily gain weight, and then we put them on the Western diet version of the isoleucine diet, and also treat them with rapamycin. You can see the mouse sort of half their size in a month. And if you inject rapamycin, you can see that it also blunts the weight loss effect of the diet. So the effect size is not as big as you can see here, but it clearly reached a different plateau. And if you are just paying attention to the details here, rapamycin by itself causes a significant amount of weight loss in these animals. So this was actually, mechanism was figured out a little bit earlier this year, and that was the leptin sensitivity story by Jeffrey Friedman from Rockefeller. That's sort of a different story. So we took these animals, we did a glucose tolerance test. What you see here is that in the context of the Western diet, isoleucine restriction is able to improve the glucose tolerance of these animals. So you see that when the dietary context is different, the diet-drug interaction also changes. Okay, so how does this work? That's obviously the big question. So I've shown you before that the low protein diet extends lifespan. So it's been shown that if you knock out this enzyme, or there's this hormone called FGF21, you can abolish the lifespan extension effect of the low protein diet, and also some of the metabolic effects. So FGF21 is predominantly a liver hormone that is known to signal to many parts of the brain, which then activates sympathetic nervous system to increase energy expenditure in the fat depots. And to make this more complex, many organs can express FGF21, and also FGF21 can signal directly to the fat. So we sort of understand that what FGF21 does overall, but we try to understand what goes on before the expression of FGF21. So animals that are fed the low isoleucine diet have a high level of FGF21, both in circulating amount and also in gene expression levels. And if you treat them with rapamycin, this is completely inhibited. So now we have sort of this working model of how things work with isoleucine restriction, where the low isoleucine diet goes through something that's inhibited by RAPA, induces FGF21, which leads to energy expenditure increase. Okay. So as I mentioned before, we know how rapamycin works. And to differentiate whether or not it's mTOR complex 1 or complex 2 that's mediating the effects of FGF21 expression, we turn to genetic models to knock out either Richter for complex 2 knockout or Raptor for complex 1 knockout. And then we put the animals on isoleucine restriction. So this is the first experiment. We cross Richter flux animals with albumin-crete, which means that the animals are knocked out for liver Richter, which in turn prevent the formation of complex 2. So these are complex 2 knockouts. And we ask the question of whether or not this is sufficient to recapitulate the effect of rapamycin. So I'm showing you this first, so obviously it didn't work. You can see the blue line here completely overlapping with the orange, which means that the knockout and the wild type have the same phenotype, the same body weight, the same energy expenditure. And when you look at their glucose tolerance, they also receive no change from the loss of complex 2 in the liver. So the next step, we look at complex 1 knockouts. So to approach this, we did a little bit of different methodology. The way we did it was to use adeno-associated virus to express the CRE in the liver through retroorbital injection in the Raptor flux animals. So we can make an animal that is an adult and to knock out complex 1. And we ask the same question. So in this case, you can see that the energy expenditure is blunted by the loss of liver mTOR complex 1. And most importantly, FGF21 expression is also blocked or blunted by the loss of liver complex 1. So this leads to our working model, which shows that the low isoleucine dad is able to express FGF21, but you require liver mTOR complex 1, not complex 2, which we're able to dissect out using either rapamycin or the adeno-associated virus. So there are a lot of data that was sort of very interesting that popped out, and you sort of noticed that we switched the virus model and we didn't keep using the animal model with the albumin Cree. So we did do that, and let me explain what happened. So this is an animal that was injected with Cree, and we're able to do Western blot from the one for every week after it was injected with Cree. And you can see initially at the bottom of the red box that there is a robust Cree expression on the first couple of weeks, but every week it gets weaker and weaker. And when you reach week 5, essentially you have a wild-type mouse, and also raptor comes back. So initially we did the albumin Cree cross with raptor flux, and what I realized was that the liver cells that are knocked out eventually gets outcompeted by the liver cells that are not knocked out, and then you just end up with a wild-type animal. So I didn't have as many panels of data because you only have really 2 weeks or so to do your experiments. And that sort of resulted in some very funny data that we sort of don't have an explanation for yet. For example, the body weight of these knockout animals actually continue to go down. So they lose more body weight than the mouse that are wild type, despite the loss of FGF21. They also have slightly decrease in food intake, and their glucose tolerance is actually better than the wild type animals. So the knockout model and both genetic models are liver knockouts. And we think that rapamycin, because it goes through the entire body and also given a high dose chronically, they are probably targets perhaps in the brain or in the adipose tissue that are mediating this discrepancy we see between the drug and the diet and the drug and the genetic model. Okay, so that's sort of the data I wanted to show you guys. Just to sum it up, we have this very interesting diet-drug interaction between isoleucine restriction and rapamycin. But the dietary context, so if the mouse was fat versus the mouse were skinny to begin with, that sort of alters the interaction. And this interaction effect requires liver mTOR complex 1 and not complex 2. Okay, so I'd like to thank some people before I take questions. I want to thank all the past and present members of Lemming Lab, and especially my students. The present ones are Brent, Ted, and Melissa. I also like to thank Judy and Brian for help with the protocol of the viruses, and also our collaborators. Also my funding sources from the NIA, as well as the Wisconsin ADRC. All right, thank you. So I'll take any questions you guys have. Thank you. Yes.
On the first part, where you decrease the and have some effect, lots of lifespan. So when is it possible that you are also decreasing protein metabolism in general? Because if you decrease one amino acid, like that's necessary for whatever protein that you're going to make. So if you don't have these one amino acid, in fact, the building blocks for every other protein that you're going to make.
Mm-hmm.
And in that case, you might have an increase in levels of other amino acids, which some of them are toxic, and would have other problems, for example, for the kidney to excrete the nitrogen in our stuff. Is it possible? My question, sorry, it's too long. But the idea is it possible that you can accomplish a similar result? You're talking about BCAA.
Mhm.
So I guess you can accomplish similar results with leucine and you show some of that. But also if you decrease other amino acids specifically.
Yeah.
In proportion. So for example, because isoleucine will not be very present on many foods. So as you're dealing with different foods, you probably have to decrease other amino acids more than you decrease isoleucine to get the same result. But is it possible that you get similar results with other amino acids, even not branched chain amino
Oh,
acids?
yeah, definitely. So that's sort of, I guess like the main one I presented, it was the isoleucine one, because this was the one we found to have the biggest magnitude of effect. So I guess the paper that's being reviewed right now, it's the valine story. So I show you that valine also improved the glucose tolerance by a little bit. And it turns out if you're able to feed the mouse lifelong a low valine diet, they also extend lifespan. So that's work done by Mariah, and that's currently under review. We also, I mean, throughout the years that we've gone through every single amino acid, right? Every diet. So the non-essential ones, we can take out completely, and most of them are okay. But we're also studying a lot of them. Some of them have like sort of special effects, involvement with different metabolic pathways, such as cysteine and methionine, because those are like sulfur-dependent amino acids. So those are a little special, but most of the non-essentials, like the mouse don't do anything. But the essential ones, so the methionine has been studied for a really long time, and that's sort of a sulfur restriction. If you were to decrease that, animals also extends lifespan. Yeah, I think that's about it. So it was pretty surprising that the leucine restriction didn't do anything in our hands. If you decrease leucine to like 0, eventually the mouse, it will look like the mouse had nothing wrong with it, and then it just dies. Yeah, so to answer your question, yes, we tried the restriction of everything, everything we can get our hands on, and we sort of did it in a very systemic manner approach. Yeah.
Maybe following that, so it looks like the low isoleucine has a stronger effect than low all branched chain amino acid.
Yes.
Which suggests that maybe having leucine and the absence of isoleucine is important.
Yeah.
And then connected about that, of course, leucine is supposed to be activating mTOR and how you're thinking about that.
Yeah, so we don't have an answer. So obviously we think this is like a ratio thing. The ratio of the amino acids are important, more important than that it's absent. So the body is actually very effective in buffering amino acids. So even though what we have is restricted, if you look at the circulating level, they are almost unchanged. And if you look at the tissue level, so it is decreased a little bit in tissue, but the body sort of compensates for it. So we do think this is like a racial phenotype. Obviously, so the animals eat a little bit more than the control fed mice. And you can argue that we can do an experiment where we supplement the other ones to simulate the change in ratio. So that's sort of an approach we're gonna try to work out. Yes.
I just have a clarifying question. So you have a lot of glucose sensitivity data.
Mhm.
Like when you had low levels of lactate or low levels of glucose tolerance. And I'm just wondering if you think that's due to just generally improved metabolism, or do you think that the signaling in terms of isoleucine amino acid and in terms of glucose with M4R overlapping?
Yeah, so to answer that, so there are a couple facets, obviously. So part of it, the mouse loses weight, so they're less fat mass. So your fat is less sensitive to insulin. So it's not going to mediate. And when we do the injection, it's dose dependent based on body weight. So if you have more of that, it's fat, you're not going to be as sensitive and be more able to control your glycemia. The other answer to that is yes, the mouse are more insulin sensitive as a whole if you control for lean mass. So we have done clamps experiments where you basically hold the mouse in isoglycemia, and then you see how much 1 unit of insulin affects their ability to uptake blood sugar in like a completely artificial environment where you're just looking at how sensitive they are to insulin. So these mouse that are on the low isoleucine diet are more sensitive to insulin. So there's sort of two sides to that question. And then there's also this part where it stops glucose neogenesis of the liver. So that we haven't looked into as much, but you can sort of see it in the bumps in the blood glucose where it doesn't go up as high. That's sort of one of the first steps that we see in there. Okay. Yes.
You mentioned the low isoleucine diet mice had changes to the heart musculature.
Yes.
Okay. Did you also check for that with the mice that had the low isoleucine and the rapamycin diet?
No, I didn't. So rapamycin by itself is known to reduce sort of the size, the thickness of your heart. So it's sort of confounded in that way. And we were only looking at in the context of age mice. So that was like a separate study where they are expected to have age-related defects. So that wasn't sort of the diet drug interaction. So yeah, so that'll be very interesting to look at. So obviously rapamycin has already been proven by other labs that it have a beneficial effect. So so that'd be interesting. Yeah, thank you. Anyone else? We'll see. I don't think there's questions online, so. Okay. Yeah. No, sorry, go ahead.
Last one. I haven't had any problem with the kidney. I'm still impressed about what's going on with the other amino acids. When you decrease one, in general, have the others in excess. And you say that the body buffers, which makes sense.
Mhm.
I work with insects, so they kind of, they don't buffer.
Oh.
They try, but they do really bad. But in general, you need to screech or do something with the nitrogen of these amino acids. What?
Yeah, so these mouse, so they do pee a lot more. They pee like twice as much. We haven't seen any kidney phenotypes, anything that we've looked at. And yeah, and then there's really isn't in like our lifespan studies, they really don't have changes or like kidney failure or kidney tumor that we were able to see. What's very so in that aspect of what you were asking, I have like more experiments where we increase the protein intake. So in those mice, they really pee a lot because they have so much nitrogen, right? So I'm driving the system very hard. And hopefully maybe I'll get more phenotypes in those animals. But so far, maybe just because they only live, 3 years, I don't see any sort of detriments to their kidney function. Yeah, thank you.
Great.